So Charlotte obtained a PhD from the University of Cambridge, where she applied directed evolution to study the mechanism of catalytic promiscuity in the alkaline phosphatase superfamily. She then moved for a postdoc at the University of British Columbia in the lab of Nobuo Tokuriki, where she continued studying the mechanism of enzyme evolution, including some fascinating work on how epistasis shapes evolution trajectories. So Charlotte, please, the floor is yours.
Thank you, Anastasia, for the kind introduction. Today I'm going to talk to you about epistasis and how understanding its molecular basis can help us design better protein and understand natural evolution. And the first question that we're asking is, how do proteins evolve? But what we want to do eventually is, how do we evolve proteins? And these two questions are obviously connected. So protein evolution and engineering is the process of climbing a fitness landscape. Here in British Columbia, we love to use mountains to depict this phenomenon. But the idea is that as you accumulate mutation, you're going to climb and increase the function. So if we translate this to a 2D format, a little bit easier to comprehend, we have our starting points at the bottom, and we want to climb to the highest fitness peak. However, sometimes when we follow certain path, we can reach local maxima and get trapped in a less optimal or less fit intermediate. So the question that is interesting in this context is, how accessible are these fitness peaks? How often and why do we get trapped in these local maxima? And the answer to the why is often epistasis. But before describing epistasis, we need to understand the effect of mutations. And when we study the distribution of fitness effect, which is the ratio of deleterious, neutral, and beneficial mutations, what we see is that neutral mutations are about 30% of the total effect. A vast majority of mutations are deleterious, and only 1 to 5% are beneficial. And this has led Charles Craig to say that protein terrorism is a more suitable descriptor than protein engineering. So most mutations are deleterious. And epistasis is an added complication. So this is a tweet that I stole from Killian Campbell, that explains a good metaphor for epistasis, where sometimes you're wearing a nice shirt and nice pants, but when you combine them together, you look terrible. And this is what happens with mutations as well. So epistasis is the non-additivity of mutations. We combine mutation A and B, and it deviates from this additivity. And so, for example, you can have magnitude epistasis. That means that two mutations will have a greater effect when they are combined than individually. However, you can also have diminished effect, such as magnitude epistasis, which is a little bit below additive. Sign epistasis, when one will be negative and the other positive. And reciprocal sign, which is one of the hardest things that happens for prediction, because two positive mutations can lead to a deleterious effect when they are combined. So how prevalent is epistasis along evolutionary trajectories? With the rise of deep mutational scanning technology, we can look at pairwise combination and have a good metric for epistasis. So when we look at pairwise combination in GB1 or the PDZ domain, for example, the authors found that epistasis is about 4 to 15% of all combination. So it's not that much, right? So should we really be worried about that? When we look at adaptive trajectories, where mutations are selected by evolution, we see that contrary to this, epistasis represents about 50% of all the combinations. So along evolutionary trajectory and selection by natural evolution or in the laboratory, epistasis is much, much more prevalent. And the consequences of this is that if we go back to our fitness landscape format and we want to climb a peak, combinations of mutation that can be positive, for example, in this case, which is more dramatic, that can result in a deleterious effect. And in this case, we will just end up diving into this valley and not being able to climb this fitness peak anymore. But fitness landscape are interesting and important, but this epistasis is rooted in biophysical interaction, and it has a molecular basis, right? And this is what we study in the Tokoriki lab. So some mutations, when mutations are additive, that means they don't physically interact typically. However, when they do interact, they can create new hydrogen bonds, for example, which will enhance the function and then create this synergy. Sometimes you can have a steric clash when you introduce two mutations, and that is a rational explanation for this deleterious effect we see here. However, some combinations are a bit more complicated, and we need to look at a bigger effect. So in order to understand this, one has to consider a protein as a network and not just a blob. And so all the residues in the protein are connected. And this connectivity creates epistasis, essentially. So here we represented in this review in Current Opinion last year, the different cases, different scenarios. So in one case, you can have two mutations that interact one another and with the substrate. This is the case of PT. This is our PET enzyme in the Tokoriki lab. I'm going to talk about this protein later. But you can see here that you create new hydrogen bonding network between the residues and the ligand, and that enhances the function and creates a very strong synergy, synergetic epistatic interaction. Sometimes you can have two residues interacting, and they will result in a change of a third residue that, for example, binds a metal ion. And the shift in the position of this metal can also affect the function. Sometimes epistasis can also be relayed via a ligand. This is less documented, but Paul O'May, who used to work in Norwich in the UK, worked a lot on this terpene synthase. And then he showed that some mutations that are really far away do interact epistatically, and that's because they relay through the substrate. A little bit more cryptic is when epistasis is propagated via conformational dynamics. So you can have two very remote mutations, but these mutations will impact the dynamics of loops, for example. This is the case here in TEM metallobeta-lactamase, where the omega loop is changing its conformation, and it's affecting the function. And then, of course, at the quaternary level, you can have epistasis across subunits. And this is a beautiful work described by J. Storz in hemoglobin, where mutations in different subunits impact the allostery of the protein. So you can see there's a lot of molecular basis for epistasis, and we can study it and understand better the effect of mutations. So how does epistasis affect protein evolution? I'm going to tell you now a little bit more about a project with more research data that I've been working on and we are writing right now about how a mutational event can create this bifurcation in the landscape that I've described earlier, how one mutation can basically change the outcome of evolution. So phenotypic diversity is very prevalent in nature. In this case, I like to always start with this example from organisms. These are the wings of Drosophila described by Gompel and Prudhomme, where you can see a huge diversity of patterns and striation on the wings of the Drosophila, yet the wing carries the same function. So different solution to one function, one problem. This is also existent in protein, also less documented. But if we go back to the hemoglobin here, you can see that in various organisms, there's many shape and forms for hemoglobin to carry the same function, which is binding oxygen. But you can have anything from tetramer in human to 180 subunits in the earthworm. So how is this phenotypic diversity arising? And how can we look at this back to our fitness landscape? So imagining a trajectory from an ancestor sequence evolving step by step with different mutation all the way to a fitness peak A. The fitness is represented here, and then the genotype is in this frame. How do we know whether there are other fitness peaks in the landscape? Because if we replay evolution, and this is like the most accessible path to this peak, replaying over and over again will always bring us here. So are there other peaks in this fitness landscape? How do they look like? And more importantly, can we access them? And one way to sort of explore the landscape around this existing fitness peak is to study these bifurcation points. So Richard Lenski did this with the long-term E. coli evolution. He propagated E. coli in the laboratory for over 20 years. And after 30,000 generations, he saw the evolution of a new phenotype, the ability of E. coli to use citrates. So here we have one of these, very strong bifurcation events in an organism. And you could call this new lineage a new species, if you will. But when you are a research associate, you cannot propagate experiments for 20 years. And so one way to accelerate this phenomenon a little is to block the very first mutation that appears repeatedly in a path and see if evolution can find an alternative trajectory. So we have a good framework in the lab to study this, because in 2012, my boss, Nobuto Koiki, evolved this enzyme, PT phosphotriesterase, towards the ability to degrade an ester called 2-naphtylhexanoate. So he did a screening and selection for the ability to degrade this chemical bond here. And after 18 rounds, 18 mutations, he enhanced the function by 7 orders of magnitude. So this is how it looks like. If here we look at the esterase activity in the y-axis and the rounds of evolution, you can see that step by step, we actually enhance this function quite dramatically. This trajectory is called the arginine trajectory because the very first mutation that occurs is arginine at position 254. So our question is, what happens if we block this first mutation by mutating it to an alternative amino acid, such as serine? So serine is interesting because it's only one step away in this neutral network. It has virtually the same function as the wild type, and it cannot mutate back to arginine unless you have 2 mutations in the codon. So we started the evolution from serine 254, and we evolved it under the exact same conditions over several rounds. And you can see when we reached round 8, we got stuck. We reached a dead end. So we started to dive into the molecular basis. Why did we get stuck so early? And what kind of mutations did we get? So when we look at the distribution of the mutation, you can see in purple. So by the way, for the rest of the talk, purple will be this arginine trajectory, and blue is anything that pertains to the serine trajectory. So in purple, you can see that the arginine trajectory mutations are scattered all over the protein, the active site being here, represented by these 2 zinc ions. And you can see that the blue mutations are sort of more localized in this part of the protein. The thing that was striking at first is that 3 identical loci are targeted along both trajectories. So position 306, 271, and 233 were mutated in both trajectories. The thing that's interesting is that they were mutated to different amino acids. So that was a bit striking. So we decided to study, whether there is a reason for these different amino acids at these positions. So first, we already had a lot of structures from Colin Jackson, our collaborator at the University of Canberra, for the arginine trajectory. And for this project, we crystallized every single protein along the trajectory. And the first striking thing that we noticed is that the substrate in the arginine trajectory binds in this cavity. It's a very nice enzyme substrate complementarity, but you can see that in the serine most evolved variant, the substrate binds in a completely different orientation, whereby the leaving group is rotated by about 90 degrees and seems to be in a pocket that's way more solvent exposed. So that was quite surprising, yet this is catalytic. So different solution for the same function, which is the hydrolysis of this ester. So we looked a little bit at these 4 positions that are mutated in both trajectories. And in the arginine trajectory, what we observe is a very robust hydrogen bonding network between these 3 positions, 233 and 254 and 271, such that the arginine here stays in an upward conformation that enables pi cation interactions and stabilization of the leaving group here. So it promotes a very efficient catalysis. However, in the serine trajectory, you can see that the amino acids that have been selected do not enable hydrogen bonding network in any shape or form. And also you can see that this loop 7, which is really, formed here and capping the active sites, is not present in the serine structures because it's so dynamic that we can't actually assign any residues in this area. So clearly, two different strategies here again. And you can see that in one case, we are building a strong intramolecular network, and in the other case, we are destroying it. Then we did a molecular dynamics simulation with Silvia Osuna in the University of Girona. And what she showed was that the loop 7 was, in the most evolved variant, R18, pretty stable. However, in the serine variant, this loop was really all over the place, adopting a broad range of conformation. With Colin Jackson, we looked more at ensemble refinement over all the structures that we had. And what Colin showed is that loop 7 is dynamic, but more stable in the arginine variant than in the serine variant, where we model the B factor here, deviation, you can see that can go up to 8 angstrom. So almost twice more mobile, if we can use this metric. So clearly, the adaptation along the serine trajectory results in different molecular interaction, and that creates more dynamics of this loop 7. So then we looked at why these mutations were selected, why different amino acids were selected along the arginine and the serine trajectory. And one way to do this is to mutate the residues back to the wild type and estimate the contribution of this residue to the function. So when we mutate in the most evolved arginine variant, we mutate the 254 residue, which is one of the key residue, back to histidine. You can see that the function decreases by 1,000-fold, which is nearly back to the ancestor level. Position 233, 271, and 306 are more modest decrease, but they still decrease by about 10-fold. And when you combine mutation 254, 233, 271, and 306 in the arginine most evolved variant, we completely annihilate the function. So we can see that this 4 position actually recapitulates the whole enhancement that we've seen along the trajectory. Along the serine trajectory, however, when we do the same back to wild type mutations, you can see that the effect is much less dramatic, about 10-fold, 10 to 100-fold decrease for each individual mutation, as well as combined. So clearly, these residues are not so important for the function in this new trajectory. Another thing that we can do is look at transition. So what happens if we put the amino acid selecting along the serine trajectory into the arginine most evolved variant and reciprocally? And this kind of tells us, whether these amino acids were selected because of the initial mutation and they have a role together, or if they can be transplanted across and they are not so important. And what you can see very dramatically in the arginine trajectory, when we put a serine, an alanine, a histidine, or all of these 3 mutations combined, we completely kill the function, about 1,000-fold decrease just for the serine, and then the triplets decreases the function completely. However, in the serine trajectory, if we put an arginine, a glutamine, or phenylalanine, we have almost a neutral effect. And again, this confirms that these positions are no longer relevant for catalysis. So then we looked at all the stepwise molecular effects that contribute to these differences between these 2 trajectories. And for this, Colin Jackson and Joe Kamarski at the University of Canberra did ensemble refinement on every crystal structure along the serine trajectory and selected variants along the arginine trajectory. And without going too much into the detail, what they see is that the loop 7 in the arginine trajectory goes through a phase of destabilization and then becomes very stable in the most evolved variant. Whereas in the serine trajectory, this loop becomes increasingly mobile, such that from round 3 onwards, we can no longer see it. If we plot only the average normalized B factor for the loop 7, you can see that for the serine trajectory, it keeps increasing, whereas for the arginine, it goes through this stabilization. Same for the arginine 254 sidechain. You can see that in the arginine trajectory, this residue becomes more stabilized because we are forming this hydrogen bonding network. However, in the serine trajectory, it's just all over the place. And again, that confirms that in one case, we are stabilizing interactions and we're stabilizing the loop gradually along the trajectory, whereas in the serine trajectory, this position no longer contribute to the function, and so they gradually become more dynamic and under less constraints. So we looked at this loop 7 a bit more in detail. And one way to study the effect of this loop 7 in each variant and in a gradual fashion is to remove the loop completely from every variant along the arginine trajectory and the serine trajectory. So when we do that, where here we represent the fold difference in function between the original variant and the same variant without the loop 7, you can see that removing the loop in the wild type is neutral for the function. But as you remove the loop progressively along the trajectory, this becomes increasingly detrimental. And that tells us that the loop 7, over the course of the evolution, became gradually stabilized and gradually important for the function. However, you can see that in the serine trajectory, you can remove the loop. It's slightly deleterious at the beginning, but becomes completely neutral. And on the other end, this tells us that loop 7 is no longer important for the function. We can also gradually look at the effect of this transition that I showed you earlier, which is what happens if we put a serine in every arginine variant along the trajectory and an arginine in the serine trajectory. And you can see that this really mimics the loop 7 pattern in the sense that when we mutate the arginine to a serine in the wild type, it's neutral and it gradually becomes deleterious. However, in the serine trajectory, adding an arginine becomes completely neutral over time because there is no other residue to stabilize it and it no longer contributes to the function. So I hope that I convinced you that these two enzyme forms now are very different. They are phenotypically different and they perform catalysis in a different way. And I like to take this image of a Jenga tower because it shows us that once we set up an initial mutation, which is this bifurcation event, we start to accumulate mutation, we pile up the mutations, and these mutations interact with this initial mutation and they form bonds. And once this is happening, you can no longer remove the little brick at the base, which is the arginine mutation, for example, because this is going to end up collapsing the whole network. And so finally, we wanted to look a little bit more at epistasis between this allele. So we can create a local fitness landscape, which is very, very primitive because we're only looking at 3 positions and 3 different amino acids, but it gives us an idea of the resolution around these 3 combinations. So what happens is that we can combine histidine at position 254, aspartic acid at position 233, and leucine at 271. That's the wild type combination of this triplet. We can do the same. REF is what we see in the most evolved arginine trajectory, and SAH is the triplet in the most evolved serine trajectory. But we can also make all the combinations in between, and that gives us hybrid mutants, which encompass mutation from the arginine and serine trajectory. And that kind of tells us whether these positions were selected specifically on the trajectory or whether other possibilities could have occurred. So when we do this, this combinatorial mutation that result in 27 variants. So we can measure their fitness for the aryl esterase activity. Each genotype is one of these little beads, and you can see in purple, these are mutations that, genotype that contain mutations from the arginine trajectory. The gray ones are the wild type amino acids, and the blue ones are from the serine trajectory. So it's a little bit hard to deconvolute quickly like this, but the take-home message from this is that first, the trajectory that evolution selected is the fastest path to the highest fitness. So evolution does find the fastest path, even in the laboratory. And the other thing that we see is that besides the genotype that were selected, all the hybrid genotypes are actually less fit than the ones that only have arginine mutations or serine mutations. So that means that these mutations were really selected together, and they are not the result of random selection. And when we study case by case, you can see that when we combine two arginine mutations, two arginine trajectory mutations, we have very strong epistasis. However, when we look at intermediate variants, for example, combining arginine and alanine, we actually have a negative epistasis. And this is true for all of the hybrid genotypes. So clearly, these two paths were selected individually. And as we progress in evolution and with mutational step, we are diverging such that now the most evolved genotypes are located on different fitness peaks. So we've showed that two distinct molecular mechanisms have evolved. We've shown that this is a very gradual phenomenon, that epistasis sets in very progressively. And we've explained this divergence with genetic incompatibility that arises from epistasis. And I just want to finish with some suggestions to improve engineering, because we can see that epistasis is complicated and navigating this landscape is a difficult task. And we've borrowed a lot of tricks from nature to do directed evolution in the laboratory. We use computational biology, but perhaps we've missed a few things from nature that we haven't quite implemented. And as Marcel Proust, a French writer, said, the real voyage of discovery consists not in seeking new landscape, but in having new eyes. And this is what we are working on right now in the lab. Perhaps we can keep the landscape, but explore it in a different way. One way would be to navigate the landscape using different mutations. For example, insertions and deletions are very dramatic mutational steps that enable you to jump in the fitness landscape. And we've just published a review last year on this in biochemistry. And then another way is to perhaps start the voyage from a different location that is an alternative starting point within the fitness landscape. And we just published this in Trends in Biochemical Science. It's a review last Friday. So if you want to read more about this, you can check this out. And yeah, I hope I convinced you that epistasis and higher order epistasis is prevalent, that it can influence evolutionary pathways and outcome, and that perhaps there are other ways to circumvent these pitfalls. And I want to thank our collaborator, the Jackson Lab at INU, and then Eleanor Campbell and Joe Kazmarski, who did fantastic work in crystallizing all these variants, and then the Osuna and Fexas Lab at the University of Girona, who did some molecular dynamics simulation and short pass mutation analysis, which I haven't shown today. And then this is our lab at the University of British Columbia. I want to thank Nobu, who is the best boss that you can have. And then Carol Buda. We are writing a lot of reviews and papers on epistasis together at the moment. And yeah, that's it. Thank you.
Thank you very much, Charlotte. That was really interesting. One question. So how far away are the mutations you studied from the ligand? Are they on the binding site or far away from it?
Yep. Oh, maybe I should share again. Sorry. Yeah. So overall, the mutations are close and far. So in the arginine trajectory, Nobu found that at the beginning of evolution, he tends to accumulate mutation very close to the binding site. And as I've showed you here, these mutations actually directly interact with the substrate. However, he had about 18 mutations. So as evolution progressed, he started to fix mutations a little bit more remotely. We know that some of these mutations contribute to dynamics, some contribute to stability. And so, yeah, I would say that the mutations that directly enhance the function are in contact or close contact with the substrate in this case.
I was wondering, so it was really interesting, your observation about loop dynamics, how they shape the trajectories. And I was wondering if to your opinion, the dynamics of the loop has a role to play in
Yeah.
in that trajectory.
Yeah, and you were studying that in Florian's lab with me as well, so you know all about it. Yeah, so it's very striking. So I didn't talk about it at all, but the phosphotriesterase, this PT, its initial function is to degrade a pesticide called paraoxone. It's a very fascinating case of xenobiotic evolution in nature. So this enzyme evolved in Pseudomonas to degrade this pesticide. And in the native function, in order to bind the paraoxone in this orientation, the loop has to open and close. So Colin Jackson has published a paper in PNAS about this. So the dynamics of the loop for the native function is very important. However, for the promiscuous function that we evolved, the dynamics seems to be detrimental. And this is why over the course of evolution on the Arginine trajectory, this loop got progressively stabilized and anchored through this 271 mutation. So in that case, it was very important. In the serine trajectory, the loop is all over the place. It's extremely dynamic. We even found a new dimer form where the loop forms a swap dimer with another monomer, which doesn't exist at all in PTE. So in that case, it doesn't seem to be relevant. So again, suggesting that it's two different molecular solutions.
Interesting. Yeah, so we have a few more questions. One from Christopher Johansson. So thanks, great talk. When you say epistasis is abundant, have you looked for something like threshold epistasis, where substitutions are additive in a linear related dimension, and the sum may be transformed to the observed fitness?
Yeah, so I think you refer to non-specific epistasis probably. So yeah, in this case, we really stick to specific epistasis. That is a mutation that directly contribute to a biophysical parameter. So we look at how two mutations will impact the kcat over km, the ki. So these are easier to study because the function is a direct result of the mutations. However, yeah, lots of people look at non-specific epistasis. In those cases, for example, for antibiotic resistance, it's very common that two mutations will impact the fitness, so the ability of the bacteria to survive the antibiotic, but these are actually they deviate from linearity with the biochemical parameter. So we haven't looked at this in this specific case because we are more interested in direct effect, not so much stability extra. Yeah.
Thank you. Yeah, so another question. So amazing talk, thanks. Can you give some detail on what happened during DMD simulations? As for the serine trajectory variant, did you observe a detachment of the ligand?
Yeah, that's interesting. Yeah. So I didn't show the substrate here. So we have simulation as well with the substrate. It's quite interesting. Yeah. For the arginine, so the substrate doesn't bind very well in the wild type. So we struggle to get a stable binding there. But in the most evolved variants, we see that at the beginning, they tend to roam around the metal ions, and then in the arginine trajectory, more quickly adopts the final optimal binding position. However, in the serine trajectory, it tends to bind a little bit in the arginine binding mode, and then ends up stabilizing in what we see in the crystal structure. So clearly, there could be two binding modes for a little bit. Yeah, this is actually quite interesting. And another thing that I didn't mention is that the binding mode of the serine trajectory that we see here resembles what we think is the native function of this PTE enzyme, which is lactonase. So binding of lactones. Lactones actually bind in this pocket. So it looks like as evolution happened in nature, this pocket was progressively closed, and then now we sort of reopened it. So multiple binding modes, yeah. But it is stable in the final variant, yeah.
Thank you. I had also one last question. So how are these trajectories partition in natural evolution. And so, and also, I mean, you change the specificity, but are you also changing the underlying promiscuous mutation and promiscuous activities between the two trajectories?
Yep. I actually have a slide for this. So we looked at promiscuity because this is obviously my first love from the Oldfelder lab in Cambridge. And what we see is that it's quite different between the serine and the arginine trajectory. So in the arginine trajectory, the esterase function really, is like the one being selected, and every other function seems to slowly trade off and disappear. So whether it's lactonase, we try 2 different lactones, which is supposed to be the ancestral function, and then 2 different types of organophosphate, paraoxone, which is the native substrate, and parathion, which is slightly different. However, in the serine trajectory, you can see that the triesters trade off pretty quickly, but the lactones kind of stay neutral, doesn't change that much. And again, that tends to confirm the fact that in this trajectory, we kept the lactone binding pocket active. And so the substrate is kind of happy here. Whereas in the arginine trajectory, as we progress, the lactones can no longer bind. So there seem to be different promiscuous activity arising from the different, repurposing of the active sites. Yeah.
Thank you. Thank you. We have another question that just came in, so I'm going to ask right away. So, very interesting talk. It was very clear and captivating. Since the first mutation can influence greatly the outcome of the final solution after directed evolution, how can you choose rationally which residue to mutate first and to what amino acid?
Yes, that's a very good question. So this is what we explored in this review in Trends in Biochemical Science that we just published. It's really hard to predict, right? Which protein is going to give you a good answer. So you can look at a few parameters. What we show in this review is that there are some things that you can measure at the beginning. For example, stability, level of promiscuous function. The fold of a protein can give you an idea of which one is the better starting point. Down to the single mutation is quite tricky if you don't know how your new substrate is going to bind. So it's probably a matter of taking several starting points, I would say. For example, for the industry, I think that would be something relevant. Yeah.
Thank you so much for this very interesting discussion. So, so unfortunately, we have.
I have a question, if it's
yes.
possible.
Yeah.
I was raising the hand. Sorry. Charlotte, this talk was really amazing. I make use of my privilege of being able to open my mic. I was wondering, from your fitness landscapes, I mean, it seems like the arginine trajectory, it's in many dimensions like preferred. But to me, maybe I'm understanding it incorrectly, it seems a little bit more fragile, because when you look at the mutation landscapes and when you check how important the different amino acids are, there's a huge additive effect in the arginine trajectory. So it seems to me it's more fragile in terms of if there are, spontaneous mutations over the trajectory, you can just destroy it before you reach the full trajectory. Is that something you have explored, or is it completely wrong from my side? I don't know.
No, it's an interesting observation. I don't know if I would say fragile, but so okay, I can answer in 2 ways. So one thing is that when you look at this network that was built here, you have 3 residues that interact in order to keep this arginine up. And I didn't really talk about it, but this arginine in the wild type has 2 conformations. One flat, which sterically clashes with the substrate, and one up, which lets the substrate bind. And what evolution does is to freeze out the flat one and to enhance the upward conformation. So you can see that 3 residues, including this loop, keep this residue up. So you can see that here in the most evolved variant, when you mutate residue 233, which is very important because it's binding here, right? It's actually not that detrimental. And 271 as well. And so this tells me that once you have the 3 mutations interacting, you have kind of a backup. So Dan Tofik, who was Nobu's supervisor, talked about this catalytic backup. You know, maybe it's a bit detrimental because you don't have the optimal three-way interaction, but two mutations still maintain this position. So there is some redundancy that tends to suggest that it is not so fragile. There is kind of a backup plan here. But you're right in saying that, yeah, the peak may be not as smooth as I represent it. It could be quite rugged and a bit steep. And as soon as you step out, you do fall, essentially, because mutations are quite detrimental. I think that would be true for a lot of very evolved and derived proteins. Yeah.
Okay, thank you. Thanks a lot.